Hello everyone and welcome to this two-part lecture about personalized healthcare research in renal diseases. The overall goals of this lecture are that you can give a schematic overview of the research pipeline in rare kidney diseases, and that you know some of the data sources that are used in kidney diseases to find new biomolecular mechanisms for kidney disease. In this first part of the two-part lecture, I will be talking about examples in membranous nephropathy. This is a topic that I did my PhD dissertation on, and I applied a lot of the work that you'll be seeing in this lecture to answer questions about how to best personalize the treatment of membranous nephropathy. So first we're gonna go a little bit into a recap of the anatomy of the kidney. So this is the kidney. As you know, they're situated on the dorsal side of the body, just below the floating ribs. The kidney is made up out of a cortex, which is the outer layer, and the medulla, which is the inner layer. And then you've got also the pilum, where the prourine is collected. If we zoom in a little bit more and we look at the outer layer, the cortex, we see that most of the cortex is made up out of the glomerulus, which is sort of this entanglement of small vessels, small capillaries, situated in Bowman's capsule over here, and the rest it's covered by the convoluted tubule, proximal if it's close to the downstream of Bowman's capsule, and distal if it's passed through the medulla, the loop of Henle, into the passing by the Bowman's capsule again and into the collecting duct. For this lecture we will be mostly focusing on the glomerulus, because that's the part that is affected by membranous nephropathy, and most notably the cells in the glomerulus called podocytes. There are more cells in the glomerulus, but podocytes are really the characteristic cells of the glomerulus. And you can see a scanning electron microscopy picture of one of these podocytes, where you've got the main cell body, and there are these protrusions or dendrites going from it, and you can see that here the podocyte really neatly wraps around one of these capillaries of the Bowman's in the Bowman space, in the Bowman's capsule better, and there are these what are called foot processes emanating from the dendrites of this cell, and these are finely interwoven and create together the slit diaphragm. And if we zoom in a little bit more, we can see a schematic overview of this filtration barrier, where in blue are the foot processes of the podocyte, in purple is the glomerular basement membrane, and on the inside, on the blood side, you've got this fenestrated, so it's fenestrated capillaries which have pores. And actually there's another layer even within this, and this is called, and it's shown here, the nice sugary wig, it's called the glycocalyx. So actually there's four layers. We've got the glycocalyx, the fenestrate, the GBM, and ultimately the podocytes with the slit diaphragm. And it's these podocytes with some of the receptors here that are affected in membranous nephropathy. On a renal biopsy, this is what the membranous nephropathy would look like. So, and it's where the name originates. You can see here in a silver staining, the GBM has been stained, and it's dark here, and you can see sort of these spikes coming from the like a spiked membrane. And what's actually going on here, if you see it in a little bit more detail, if you color it with immunofluorescence, is that all along this glomerular basement membrane it lights up because of IgG4 deposits. And if we zoom in a little bit more with the electron microscope, we can see that these deposits form this bead pattern with spikes in between. So that explains the spikes that we saw on the microscopic image. So what is happening here? What is happening is that there's a antigen on the podocyte. In 70% of the cases, this antigen is called the phospholipase A2 receptor. And this receptor is targeted in an autoimmune fashion by the patient's own immune system, where IgG targeting the phospholipase A2 receptor becomes adheres to the receptor, and it starts activating also the complement system. And as a result, the nice food processes that you saw in the picture of the podocyte, they start to dephase, is what's called, or efface better. And the nice food processes, they just completely go away, and the slit diaphragm is lost. And as a result, the capacity of the kidney to make sure that you only filter blood and or water and small molecules is lost, and a lot of protein starts coming through this filter, starts coming through the GBM and passes the podocyte, which has lost the slit diaphragm. And as a result, people on average lose 8 grams of protein per day, but it can be as high as 20 grams per day is what I actually saw during my research. And one of the main proteins is albumin, and this is an important protein that makes sure that there's enough fluid in our blood vessels. And as the concentration of albumin is normally 40 grams per liter, but in these patients it's even less than 20 grams per liter, so less than half of the normal concentration, which means that blood or fluid starts going out of the blood vessels and into the tissues, and there's a massive edema. People start building up fluid in their legs and in their face, and in addition they've got an increased chance of pulmonary embolism and deep venous thrombosis. So, and in particular these pulmonary embolisms are very severe and can have fatal consequences. So it's a life-threatening disease. If we look at a little bit more of the scientific history of membranous nephropathy, already in the halfway 20th century, there was a discovery in a rat model. It's called Hayman's nephritis, where these rats had a very similar disease compared to the humans. In fact, in this rat model, antibodies versus maglin, a transporter protein on the rat podocyte, were formed. However, there is no maglin on the human podocyte, and there was no maglin shown in the deposits of human biopsies. So therefore, for decades, the podocyte antigen was unknown. Despite that, it was quite clear that we were dealing with an autoimmune disease, so treatment was possible. And one of the first treatments was with chlorambucil and later cyclophosphamide. Both are cytostatics which were used previously in hematology to deal with leukemia. And these molecules, they've got an interesting history. So as you can see on the left hand side here, cyclophosphamide is converted into phosphamide mustard in the body. Now, phosphamide mustard is the active component that actually is a cytotoxic component. And this was discovered, in fact, at the start of the 20th century. In the First World War, in fact, a lot of mustard gas was deployed at the Western Front, particularly in Belgium and northern France. And soldiers who survived the gas attacks after going back to their homes seemed to be more susceptible to infections. And so, late 1940s, the mustard derivatives were first deployed to suppress the immune system in the case of leukemia. This phosphamide mustard is an alkylating agent. That means that it makes has cross-links and breaks in human DNA. And as cells start to divide, the DNA is checked, and a cell that is severely damaged will go into apoptosis. So in particularly fast dividing cells, such as white blood cells in the bone marrow, which are T cells but also B cells, are more susceptible to this drug. It also explains why some people who take high doses of this drug and chemotherapy lose their hair, for instance, because those are also rapidly dividing cells who make sure that the hair is growing. And Professor Claudio Ponticelli, he's an Italian, in the 1980s first successfully used this drug in one of the few randomized trials in patients with membranous nephropathy, and the results are shown on the right, where the untreated patients, 60% of them ultimately needed dialysis, whereas in patients who were treated only, 40% need dialysis, 6% survived, and in the in the treated patients, only 8% needed dialysis, so 92% survived with their kidneys more or less intact, which is a big step up. But we're not there yet, and I'll dive a little bit deeper into this later on. So, one of the most important downsides to cyclophosphamide is that during its conversion in the liver to phosphamide mustard, also another substance is created, which is called acrolein. And acrolein is it's very toxic, and it's rapidly excreted by the kidney and starts building up its concentration in the bladder. And at high concentrations, acrolein is a very potent oncogenic exposure. So not only patients with membranous, but also patients with rheumatoid arthritis, non-Hodgkin lymphoma were treated with very high dosage, much higher than membranous nephropathy actually, since halfway the 20th century. And in these patients with rheumatoid arthritis, in particular, bladder cancer was occurring much more than expected. And as a result, the patients and treating physicians who were treating patients with membranous nephropathy had to pick between two evils, either risk dialysis or risk cancer. And if we look at the control group in the Ponticelli trial, we see that approximately half of the patients with an active membranous nephropathy will have progressive renal damage and ultimately need dialysis. However, in the other half, the disease goes into what we call spontaneous remission. It becomes quiet by itself. And so this leaves us with a scientific question where, okay, what if we can discern between people who will have progression and people who have a spontaneous remission at an early stage? Then we can personalize the treatment. And that was actually the goal of my PhD thesis. So at the end of the 1980s, there was a small randomized study in Nijmegen performed by my PhD supervisors, Jacques Retzels, and they did a study to the effect of prednisone in progressive membranous nephropathy, and prednisone turned out not to be effective. It was a failed study. However, the study enabled Jacques Retzels to do measurements about the excretion of urinary beta-2 microglobulin, which is a marker for tubular damage. Remember the renal tubule? It's actually mostly secreted in the proximal tubule, so immediately after the pro-urine has passed through Bowman's capsule, it goes into the proximal tubule, and there beta-2 microglobulin is excreted if the tubule is damaged. And as you see, there is actually, here we have a plot where we can see that the level of serum albumin does not really make a discernible between the people with a stable kidney function in group 1 and people with progressive renal disease in group 2. Neither does proteinuria. However, if we look at beta-2 microglobulin excretion, in fact, the people who show progressive kidney function loss appear to be having much higher concentrations of beta-2 microglobulin excretion. And this, mind you, is a log scale for beta-2. So they have much higher concentration compared to the people who go into spontaneous remission. So aha, this might be the biomarker we're looking for. And based on this figure, a cutoff value of 500 nanograms was chosen, approximately over here. And if we, in a later study, this was validated. And if we look at the Kaplan-Meier survival curve for which shows the survival with a relatively normal kidney function, then we can see that actually only 2 people in the group with a low beta-2 microglobulin excretion showed progression in a little over 5 years, whereas almost all of the people with a very high excretion showed progression. So this was actually one of the first markers to be discovered to make a early distinction between people who progress and don't progress and base treatment on. So this is one of the earliest examples of a more personalized treatment. And after this success, patients from all of the east of the Netherlands, from as far north as Zwolle, Enschede, all the way to as far west as Roosendaal, to as far south as Venlo, were referred to Nijmegen to have this biomarker measured and to get a treatment advice. So based on this, we at Radboud UMC created a restrictive therapeutic regimen based on beta-2 microglobulin, where people are diagnosed with membranous nephropathy. We determine the risk of progression, and then based on the low-risk patients, we give supportive therapy, which is blood pressure monitoring and a watchful waiting therapy. And the high-risk patients we treated with cyclophosphamide. And as part of my PhD thesis, I did research on medical records and showed that almost 20 years later, we had 250 patients treated according to this regimen, and we reported the results. And if we compare to in blue and in black the original trial by Ponticelli, we can actually see that this in red shown the restrictive treatment strategy is almost as good as treating all patients, but saving half the patients this increased risk of cancer. So this is a big jump towards personalized medicine, but we're not there yet still. So as I told you before, halfway during the 20th century, it became clear that membranous nephropathy was an antibody-mediated autoimmune disease. And in fact, in the 1980s, so we found that the therapies which were successful in rheumatoid arthritis and leukemia could work in membranous nephropathy. And late during the 1990s, a new drug was developed in the treatment of non-Hodgkin lymphoma. This was really a game changer. This was rituximab, one of the first successful biological drugs. Nowadays, biologicals are bread and butter, really, in the treatment of internal medicine. But rituximab is one of the first monoclonal antibodies, and it's directed against the CD20 receptor present on B cells. So here, this is where rituximab intervenes. So it's given IV. These antibodies, they attach to the B cells and then activate the immune system to get rid of the B cells. As you know, B cells create antibodies, and they can differentiate into plasma cells and even create bigger amounts of antibodies. And the major advantage and the then innovative aspect of using this biological is that the mechanism of action is actually relatively clear. We know pretty well how this drug will work, whereas the exact mechanism of cyclophosphamide is sort of aspecific and kind of mysterious. We know for rituximab exactly how it works. And with that mechanism, in mind, Professor Ramuzzi, another Italian physician, started giving his patients from 2001 on treatments with rituximab instead of cyclophosphamide. And in 2010, they published their results of a cohort of 100 patients treated with rituximab. And initially, they claimed that the treatment was effective for everyone. However, because of a statistical error, eventually we found out that this was not 100% effective, but about 80% has a remission after treatment, which makes it seem slightly less effective than our treatment with cyclophosphamide, which is 95% effective. However, the big advantage of rituximab is that it has far less side effects because it's much more specific. It only targets these B cells and not every cell which is rapidly dividing. So we decided to work together and create a so-called comparative effectiveness study. And a comparative effectiveness study tries to emulate a randomized clinical trial as well as you can. So, like a clinical trial, you've got two cohorts of treated patients. One group got rituximab, the other group got cyclophosphamide. However, we didn't exactly randomize it. The only randomization, quote unquote, that occurred was that the rituximab treated patients were all Italians, born in Italy, went to the clinic of Dr. Ramuzzi and got rituximab there. Whereas the cyclophosphamide treated patients were Dutch people referred to Nijmegen and were treated with cyclophosphamide in Nijmegen. So, using statistical methods, something called propensity score matching, we matched these patients based on their confounder profile. So we made sure that on average their proteinuria at baseline was similar. We made sure that their kidney function at baseline was similar. And a whole range of confounders was introduced in these propensity scores, and they were matched, closely emulating a clinical trial, even though this is only observational registry data. And from this analysis, we were able to show that indeed rituximab is slightly less effective than cyclophosphamide, where only 70% of the included patients were in remission, whereas this is 95% for cyclophosphamide. However, on the flip side of the coin is that there are fewer severe adverse events after rituximab treatment compared to cyclophosphamide. The cyclophosphamide group, this was about 30% in 5 years, whereas it was only 20% in the rituximab group. So this is a very real-world example of how you can use intelligent study designs to emulate a randomized controlled trial in a rare kidney disease. And this can help you personalize treatment when there's very few data available. Of course, now the next question is, can we predict who will benefit from rituximab? Who are those 25% patients who do not go into remission after rituximab but do require cyclophosphamide? Like 60%, you're good with rituximab. But who are those extra patients, this difference really, who need cyclophosphamide? And who are the 5% who are basically doomed, who we have to figure out something else for? One of those biomarkers may be the latest biomarker for membranous nephropathy. So to answer the question who might benefit from rituximab, we need to dig into the physiology of membranous nephropathy. And for a long time, this podocyte antigen was unknown. However, in 2009, 2010, there were a couple of major breakthroughs. First of all, Larry Beck, who is situated in Stanford in the US, discovered by Western blots in cases and controls a 185 kilodalton protein which showed reactivity in the serum of people with membranous nephropathy. They used a targeted mass spectrometry and compared this protein to known proteins, and thereafter, they tested antibodies from the serum against these recombinant proteins, and all these 70% of these patient sera appear to be active against the phospholipase A2 receptor that I mentioned before. In fact, also when they reviewed biopsies, again, this PLA2R co-localized with the IgG deposits. So they discovered the cause, which was before then unknown, for 70% of the patients with membranous nephropathy, which was a big breakthrough. And 2 years later, the results of that were confirmed in a big genome-wide association study, which also the Radboud UMC took part in. And it turns out that people who have a polymorphism of the PLA2R gene had a 4 to 80 times bigger chance of developing membranous nephropathy. In addition, one of the human leukocyte antigen genes, HLA-GQA1, has also a big role, and the HLH is actually a very core part of the human immune system, which really drives home the message that yes, this is an autoimmune disease. And in fact, my thesis co-supervisor published this paper where we derived the first clinical way to use PLA2R. So she showed that the presence of PLA2R highly correlates with the disease activity within patients. So there's clear abundance of anti-PLA2R antibodies in nephrotic patients. When they're in remission, there's almost none. When they relapse, it comes back. And also it's well correlated with the proteinuria. So, having this information, in 2015, the group of Ramuzzi, they published an analysis of all the anti-PLA2R positive patients in whom they had a PLA2R titer at baseline. And they correlated this with the probability of remission after treatment with rituximab. And in their cohort, patients in the highest tertile of PLA2R titers appear to have a 4 times slower and lower rate of spontaneous remission. In 80% of the cases, the people with the lowest tertile respond to the treatment, compared to only 25% after 2 years in the people who have a very high titer of PLA2R. Which means that if the PLA titer is as high as 200 RU per ml, the chance of remission is far, far lower than the 50% of 2.5 years. And perhaps these are the patients that we need to select for cyclophosphamide treatment. And this is actually something that we're looking into right now. So to summarize, this is sort of the haphazard discovery pipeline from membranous nephropathy. This is the historical overview. So in the late 1950s, the heminephritis was discovered. It took us almost 25 years to use this and start the first randomized trial, which finished in 1995. This is also the year that beta-2 microglobulin was discovered and used to further personalize the treatment. In 2001, the rituximab cohort study started, and only like in the more recent years then we started finding the genetic information. The PLA2R was discovered. A restrictive therapy was that we compared to the rituximab therapy was only done in 2014 or 2017 actually. So this is sort of a haphazard discovery pipeline. It is up to you after this course to make sure that this haphazardness is reduced, that we've got a clear idea from the start how to personalize the treatment of disease, so it doesn't take us 50 years to get the best treatment, even if it's rare diseases. So the other point I wanted to drive home here, and I showed this in the italics, is that no research design, no technology is sacred. You need them all. Like, you need the animal models, you need the randomized trial, you need the cohort studies, you need the GWAS, you need the comparative effectiveness studies, because all of them together help piece the puzzle of the best treatment possible for people with a rare kidney disease. And third, and this is a little bit of a glimpse into the future, in the future, the EU has recognized that rare diseases affect a lot of people. Actually, if you put all of the people who have a rare disease in Europe together, you arrive at a population that is as big as the total population of Belgium, the Netherlands, and Luxembourg. So this is a lot of people with a rare disease. Rare diseases are not that rare if you think about it. However, it means that you are faced with some challenges. You don't have the numbers of patients for traditional RCTs. The disease course often is long, so it takes a long time to get hard endpoints. And also the experience that physicians have and the expertise is highly fragmented. So that's why the EU created European reference networks, and Radboud UMC is one of those centers, one of those expertise centers for rare kidney diseases, where a lot of the knowledge is brought together in a focal point and really takes charge and into creating the best personalized healthcare solutions for these people. So if you find this interesting, contact us and we may be able to get some really cool internships for you. Also, because the treatment is a little because the randomized control trial is hard to set up, we've got some other really cool designs running at our department, and one of them is the N of 1 study. First, a little bit about the diagnosis of magnesium wasting. So last week you learned all about magnesium and the renal tubule. Two important magnesium wasting disorders are Gentleman syndrome and Bartter syndrome. Most magnesium reuptake takes place in the thick ascending limb of Henle's loop and is driven by the concentration gradient there. And that gradient is built up and maintained through active sodium resorption. Important transporter proteins in these processes are shown schematically in the cell, and a defect in any of these players may cause renal magnesium wasting. In Gentleman syndrome, the cause of magnesium wasting is a mutation in the NCCT, encoded by the CL12A3 gene, whereas in Bartter syndrome, generally the cause is from mutations in the NKCC2 or with the ROMK transporter. And thiazide diuretics block the NCCT and thereby induce sodium loss and consequently increase diuresis and a reduction in blood pressure. So these blocking of the NCCT is actually something that we use therapeutically in people with a high blood pressure. And this also means that thiazide diuretics can be used to establish a clinical phenotype for patients with magnesium wasting disorders. So say you've got a patient who has a magnesium wasting and you don't know exactly what is the cause yet, or what is the appropriate phenotype. You may have done a mutation screening, but you haven't found any of the usual suspects. So what can we do? You can use a thiazide, for instance, and if the excretion of chlorine or sodium does not increase as a response to the thiazide administration, this is suggestive for a mutation that affects the NCCT. And that is something that we actually do at the Radboud UMC. We have a personalized diagnostics. One of my colleagues who recently defended her PhD thesis, Dr. Anneke Beck, showed that there was a blunted thiazide response also if mutations in other part of the cellular sodium transport arise. Moreover, a patient with mutations in the same gene such as HNF1 beta, may show differences in thyroid response. And this detailed phenotyping, in addition to genetic testing, will be needed in the future to better diagnose these patients and understand the pathophysiology of the renal tubule. And this brings us a little bit to also the treatment of the renal magnesium wasting. So this is a lot of trial and error, right? These are rare diseases. There is very little known of them. Only recently, in recent years, there's these European reference network for rare kidney disease and these expert centers. So we're still learning a lot. And one of the features, or one of the complaints that patients often have, is fatigue and muscle aches due to the magnesium wasting. And most of these patients require a lifelong treatment with high doses of magnesium salts to counteract magnesium wasting. However, sufficient replenishment is difficult because high doses of magnesium salts induce diarrhea and nausea, which aggravates the problem and not alleviates it. And there's a lack of randomized controlled trials comparing different magnesium salts because there are simply too few patients to run a trial. So Anneke Berg tried a different solution to this paucity of evidence. And this is to use a N of 1 study. And the figure shows the distribution of muscle pain scores for a single patient who tried 3 different magnesium salts. And from the distributions you can clearly see that the magnesium aspartate salt, indicated by the long dashed line, was most effective in reducing muscle pain in this patient. And what's most extraordinary about the figure, that all these curves, all these distributions, they were constructed from 2 measured data points only. So even if you have a little bit of data, with the right analysis technique, you can have these very clear and very persuasive results. And in the next 2 days, you will be learning how these studies, these N of 1 trials, are conducted, and how you can get clinically meaningful results from so few measurements. And an important part of this is Bayesian data analysis, especially in rare diseases where randomized clinical trials are not feasible. N of 1 trials can be of great value for clinical practice. And we'll leave it at that.